Right. And I think maybe we can segue to Kevin's talk. So I'll give a brief intro. Kevin trained in mathematics and bioinformatics. He previously worked in the pharmaceutical industry at Novartis Oncology before doing his PhD in molecular biology. And he recently completed his postdoctoral training with Professor Charles Swanton at the Francis Crick Institute, and he's now a group leader at University College London Cancer Institute. And so, Kevin, please take it away.
Okay, thanks, Tommy. I presume everyone can see the slides. Okay, so I'll start.
Yes.
So the title of the talk is Meta-analysis of tumor and T-cell intrinsic mechanisms of sensitization checkpoint inhibitor treatment. So just to give the motivation for this project, we had two key motivations. First of all was in a biomarker context. We wanted to understand which biomarkers validated in a large meta-analysis setting and which show potential clinical utility. And then also importantly, obviously, the broadening of checkpoint inhibitor therapy to other cancer types, classically non-immunogenic tumors, prostate tumors, breast tumors, for example, will clearly be biomarker dependent. And then the second motivation was around biological understanding. So we wanted to try to build a data set that had in-depth focus of whole exome and transcriptome data, that was well powered over 1,000 patients. There's been other large scale panel sequencing efforts, but we wanted to sort of build something that would allow more exploration of the biology of IO response and explore things like differences in response mechanisms across histologies. So this is the work we undertook and published earlier this year. We published a cohort of over 1,000 patients across 8 different solid tumor types. This is all data we collated from other published studies from other industry trials or other academic trials. So it's a data aggregation based approach. But we have worked hard to make the data all comparable and harmonized. All the data sets you can see here is an overview of the study design. We take all the data sets as raw sequencing reads, reprocess them through a standardized bioinformatics pipeline to ensure it's comparable. We also harmonized the clinical response labels under the RESIST criteria with a defined outcome metric of radiological response, so CR/PR versus SDPD under the RESIST criteria. And then our pipeline produces a standard set of outputs, the obvious things like germline mutations, somatic mutations, copy number alterations, neoantigen cores and HLA types, and a variety of RNA expression signatures. And then in this study, we then, as I said, wanted to do a systematic review of previous biomarkers from the literature. And I'll show the results in a second. And then we wanted to assess the clinical utility of biomarkers either together or combined as a multivariable predictor of checkpoint inhibitor response. And then with this bigger data set to see what discovery analysis could be conducted to find new and not previously reported biomarkers. And just to mention, we're trying to maintain this as an ongoing resource. So we've got about 1,000 patients, but this is already increasing now up to around about 3,000 patients from checkpoint inhibitor trials. And we're really working hard actually try and make this as an open community resource to be able to deposit all of the math files, the copy number results, the gene expression profiles in a centralized data portal. There's been a few challenges with data access agreements and restrictions. And so we're trying to work with the original authors to come up with a framework where this data can be released, and we hope that that can be achieved, and then it will be maintained as an ongoing resource to give this data to benefit collaborators across the community. So here are the results from the meta-analysis. So we did a systematic literature review which identified in total up to about 50 different biomarkers that have been previously published as predictors of checkpoint inhibitor response. Here is in this figure is the ones we could measure in over 3/4 of all the samples. And here the blue means a biomarker that associated with high response. Red is one that associated with resistance. It was all normalized as Z-score standardized effect sizes to make the biomarkers comparable. And so what we can see, the biomarker with strongest effect size across all of the cohorts here was the number of clonal mutations in each tumor. The clonal TMB that had strongest significance and strongest effect size, and that was closely followed by TMB. It was only a marginal difference, but the clonality of mutations was slightly more significant. We found subclonal mutations to be non-significant, sort of building on prior work which suggests that the clonal mutations preferentially drive immune response. And then when we looked down in terms of measures of immune cell infiltration, we measured various signatures, and what was surprising actually was expression of a single gene. CXLC9 was the strongest predictor that outperformed any of the signatures that had been previously optimized and published, and suggesting perhaps some of those may be overfitted. And at least in this data, we found a single gene expression to be a more reliable biomarker. If we look in individual tumors, you can see here just the data for the clonal TMB measure. We can see that responders clearly have high number of clonal mutations, on average about 100 more clonal mutations in a responder versus a non-responder. What was also interesting was looking at the biomarkers that didn't replicate. So for example, the burden of somatic copy number alterations. So this has been previously published in 2007 in Science, where tumor aneuploidy was shown to correlate with a reduced response to immunotherapy. And so we investigated this biomarker in particular. You can see here some of the prior literature that shows clearly patients with a higher copy number burden have a reduced survival on checkpoint inhibitor treatment across multiple different studies here. And so we looked in more detail, and indeed we could see a separation. This was in a separate cohort, the Project Genie MSK impact samples. Indeed, patients with a higher burden of copy number alterations did have reduced survival in terms of OS, but we also saw a similar pattern for patients that didn't have immunotherapy treatment. Again, a separation in the curves. So it may be that this biomarker is more prognostic than predictive, or maybe large data sets are needed to determine that either way. We were interested to look at the variation by cancer type and by treatment type. So we had 4 different settings where we had enough samples. We had multiple independent studies published with a certain combination of histology and a treatment type, shown here. We found overall most of the biomarkers that were significant in a single histology were the same as the one significant in the pan-cancer setting. So there was a lot of homogeneity in biomarkers. We found some variation. For example, the effect size of TMB was higher in lung cancer, for example, compared to melanoma, where it was much lower effect size. We found particular biomarkers significant in the anti-CTLA-4 setting, HLA divergence measure here, which measures the diversity of our germline HLA alleles, and that, if you've got a broader set of HLA alleles, you should present a broader set of epitopes. And so that was significant just in a CTLA-4 setting, which actually may link to the mechanism of anti-CTLA-4 treatment. It's supposed to stimulate a much broader expansion of the TCR repertoire, so that may reflect specific biology for that treatment. We could look at the correlation between biomarkers. A lot of them are correlated with each other. There's redundancy between the measures, particularly within measures of mutation density, as well as measures of immune cell infiltration. But there was overall a weak correlation between these compartments. So up here it was relatively non-correlated. So that gives us some hope we can combine the biomarkers and they have independent predictive utility. We moved on in our analysis next to look at mutational signatures. We first of all did an analysis of all of the published cosmic mutational signatures, and we found several of them to be predictive of checkpoint inhibitor response, and this is after correction for tumor mutation burden, and also this was a meta-analysis, so each tumor type was treated individually. We didn't mix together tumor types in a pooled analysis. And so you can see in particular the aging signature, tobacco signature, UV signature, poly signature, and APOBEC signatures all were associated with better response to checkpoint inhibitor treatment. So this really suggests that mutation quality as well as quantity is important. And we're interested to look further at the UV signature mutations, and we noticed that UV signature correlated very strongly with dinucleotide variants. Here you can see they're both highly enriched in melanoma patients, and we saw a relatively strong correlation between the proportion of UV mutations and the number of dinucleotide variants. And it's well known that UV actually, about 10% of UV mutations are actually CC to TT mutations, so it's strongly associated with ultraviolet damage. So we wanted to further investigate why, what might be causing the preferential quality in UV signature mutations compared to other types of mutations, and whether dinucleotide variants may have any link to immunogenicity. So we compared, first of all, how the amino acids change for a normal and SNV mutation, where one nucleotide changes. Here you can see the map of how amino acids change from the reference amino acid to the alternative one. And we did the same for the dinucleotide changes, where two amino acids change within a codon, and you can see a much broader spectrum of changes, and that makes sense. I guess certain changes physically impossible. If you only change one letter in a codon, you can only go from certain combinations of amino acids, but if you change two amino acids, two nucleotides in a codon, you unlock a broader set of possible changes. We see that here. We saw for dinucleotides over around 250 unique change combinations, and that was higher, significantly higher than that for single nucleotide variants. We also saw more radical amino acid changes. This is where the whole side chain is different. A radical change was significantly more frequent for a dinucleotide variant compared to a single nucleotide variant. And what we think may be a particular factor for consideration here is the change in hydrophobicity. We saw a strong shift where dinucleotide variants were associated with an increase in epitope hydrophobicity, replacement of reference and amino acid, with the alternative amino acid being more hydrophobic. And of course, we know from viral studies that hydrophobicity is typically associated with higher rates of immunogenicity. And we could see that here again in a cancer setting, here using T-cell reactivity data from neoantigen screening exercise in gastric cancer, and we see that epitopes that were T-cell reactive had on average significantly higher rates of hydrophobicity. We moved next in the analysis to look at copy number changes. So we did an unbiased analysis across the genome, looking at the frequency of gains and losses for responders in blue and non-responders in red, and looking where these curves separate as a potential predictor of checkpoint inhibitor response. We saw the strongest separation on chromosome 9 here, particularly on chromosome 9q. Here we look by cytoband. This was significantly more frequently lost in responders than non-responders. And when we did minimal mapping of the region here, and this was quite a surprise, actually. I wasn't familiar with the TRAF2 paper that David kindly just presented, and so this came out in a very much unbiased way. The peak very sharply was around TRAF2 gene, which, as we just saw from David's talk, the strong functional data, and that was also, the top hit from their genome-wide CRISPR screen. And in terms of the direction of the biology here, we see a loss of the gene associated with better response. So again, that would fit with David's data, where loss of TRAF2 sensitized tumor cells to T-cell killing. So we hope and believe with analysis like this, we can find functionally relevant genes. We looked also in focal changes. So the prior data was broad scale copy number changes. Here we looked just at focal deep deletions, homozygous deletions, or high level amplifications. And we found cyclin D1 was significantly more frequent in non-responders than responders, associated with resistance to checkpoint inhibitor treatment. We found cyclin D1 amplifications particularly enriched in bladder cancer patients, and when we looked at mRNA level, we indeed found non-responders had significantly higher mRNA levels of cyclin D1. We were able to validate this in the MSK impact cohort data where this gene's captured, so we're able to get good quality copy number data for cyclin D1, and we found an even stronger effect size actually in the validation cohort. Here we can see hazard ratio of almost 3.5. So patients with cyclin D1 amplification really had rapid progression on checkpoint inhibitor treatment. This is in bladder cancer. This is in a control, negative control bladder cancer patients from the same study not treated with immunotherapy. There was no significant difference. And then similar for a pan-cancer cohort here of around 1,500 patients, we see significant difference in survival for CPI and then not in the non-CPI treated samples. And again, there is prior functional evidence published in 2018 in Nature, where they showed that PD-L1, cyclin D1 actually negatively regulates PD-L1. Here you can see from the pathway here, and cyclin D1 amplification downregulated PD-L1 expression. So again, that may explain the basis of that association. Higher cyclin D1 may mean lower PD-L1 levels and less chance of responding to anti-PD-L1 treatment. So the last analysis we did in the study was looking at single cell RNA sequencing data from some of our local trials, where we had patients tumor-infiltrating lymphocyte samples, and we did tetramer sort to find CD8 cells that were reactive to a clonal neoantigen. And we could compare that to tetramer negative T cells to try and see what targets were upregulated in T cells that could recognize neoantigen, and then integrate that with the patient data set to see what commonalities we could find to identify markers validated at a single cell level of resolution, as well as a bulk sequencing level. And we saw a strong outlier there. CXLC13 came up as highly significant in terms of upregulation in the tetramer positive T cells, as well as also highly upregulated in responders compared to non-responders. And again, I think that supports other evidence that's been previously published that CXLC13 is critical to driving an anti-tumor immune response. So lastly, we looked then at the clinical utility of this set of biomarkers, and we wanted to take all the significant biomarkers from the original meta-analysis and combine them together to see if we can make a multivariable predictor that would be more accurate than any one biomarker by itself. So we did some feature fitting, and we saw different features as slightly more or less important across each cancer type. But a lot of the common features were weighted most highly: clonal mutation burden, CXLC9 expression. That was commonly one of the top 2 or 3 markers for each cancer type. And then there was some variation based on the histology or the biology of each tumor type. Things like ApoBec, that's more active in bladder cancer, was weighted higher in those samples, whereas things like UV mutations came up more important in the melanoma cohorts. We did some validation. So this is again in the training cohort of samples. So these are not external samples. This is just part of our training exercise. We did 1,000 rounds of Monte Carlo sampling and held back a quarter of the samples each time to see how the predictive utility, as measured with error under the ROC curve, measure changed for this multivariable predictor using tumor mutation burden as a single biomarker that's got the strongest performance as an individual marker and is also FDA approved now for patient stratification. And so we saw across all 4 tumor types here that the multivariable predictor was significantly outperformed TMB. And then we moved to make a final model that was trained in all of the tumor types in the study, the 1,000 patients, using those set of biomarkers that were significant in the first analysis, with the feature scores here fitted. And then we validated this in 3 separate cohorts that hadn't been used in any of the training steps: a pan-cancer cohort from a keynote study here, and then a melanoma study, and then a lung cancer study. And we see across all 3 of these cohorts that the multivariable predictor had significantly better AUC performance than tumor mutation burden could achieve alone. And this is in total around 350 patients. So just to give some conclusions, we found that the anti-tumor immune response really is driven by the burden of clonal neoantigens, as well as expression of key cytokines and chemokines, including CXCL9, CXLC13. Also, we found the importance of TRAF2 that replicated following on from David's work. We also found amplification of cyclin D1 as a cause of resistance to checkpoint inhibitor treatment. So thank you very much to our funders and collaborators, and thank you very much for your attention.
Awesome. Thank you so much, Kevin. That's also a very impressive collection of work. And I guess maybe I can ask, because the work to aggregate all these data sets, I mean, I know that some of them have been released, but in order to really get, it seems like the raw data from all these clinical trials, which seems to be very jealously protected at times. I mean, I guess I wonder what you see as far as obstacles to making this kind of resource, or to making this type of collaboration happen, and getting these types of resources together to do this kind of important analysis.
Yeah, the main challenge seems to be the lawyers. I hope there's no lawyers on the talk here. I hope not to. I guess they're just doing their job. But we, I mean, we found that most, even across industry or academia, most of the scientists involved, wanted to contribute data, and certainly all the authors obviously made the data available through dbGaP or EGA, and were able to obtain the data through those channels. So the process worked well from that point of view. It's very time consuming, and it took years really to get all those data transfer agreements finished and for it all to be completed. And so to make things easier and to speed up the process for others, we want to really try and release a process version of the data that can be downloaded when you click through. Maybe you click some sort of disclaimer, but generally get the access immediately. And so again, the scientists seem supportive of that. I don't think that anybody that doesn't want it to happen from a scientific or clinical point of view. And it's just a matter of the legal agreements and how, what each company or each academic institution, what IP or what coming up with a common legal framework that everyone is happy to sign up. But we're very fortunate to have things like the Genie initiative that's already led the way, and there's framework there that could be reapplied. So we expect that it can be done.
Maybe one more question while others think. You just lost my train of thought a little bit. Give me a moment to think about it. Quan, maybe you can take it.
I'll ask related to that question. So then, are you guys able to make at least the somatic mutation calls and copy number available, for example, or that's not available?
We basically want to try and make everything available, but some of the agreements have got clauses around data derivatives. You can't release a data derivative, and what is when does something start becoming a data derivative and not be a data derivative? It's sort of things like that. The legal, we haven't quite had clarity on the legal definitions. We would hope, in other studies like TCGA, they make the somatic mutation calls available freely. So we're hoping that can be done, but based on how the DTAs are written, some of them wouldn't even allow, or it's not obvious that we can do that without getting sued. We sort of need to be careful not to get sued, first of all.
Thank you guys for doing that.
And maybe David has his hand raised.
Yeah, absolutely phenomenal work, Kevin. I think it's also a really good initiative, right? Kind of making all these data sets into one, I guess. I was just wondering, maybe going towards the future, I think you're now mainly focusing on tumor intrinsic signaling and tumor intrinsic signals. We're also thinking of maybe adding more single cell sequencing data in there to also look at, I don't know, immune cells and see how they can predict ICB response.
Yeah, no, exactly. We are starting a similar initiative to aggregate the collections of publicly available single cell sequencing data sets and harmonized processing. So that's also a project. It's some of the platform biases and differences, is a bit more difficult to deal with. So we're doing the technical work to try and check they're comparable, and then we hope to have similar benefits from a harmonized single cell sequencing data set.
And I recalled what I was going to ask earlier. As far as the multivariable prediction score, I think you touched on a really nice point, which is that TMB has been sort of approved by the FDA with maybe some controversy around that. But you're showing that this very nice predictive score that does better. But maybe one of the challenges is how to actually implement scores that are a little bit more complex than TMB, and also the level of evidence that's needed to convince a regulatory body to accept that. So I just wonder if there's any talks with probably pharma or people that are actually doing trials to try and incorporate that type of validation.
Yeah, no, that's a very good point, and we've got a prospective validation that we're working on with a national study in Denmark. That they've got a really wonderful study there, the ProTarget trial, where they've got whole exome and RNA-seq factored into their study, and they're already planning to use TMB to stratify patients. And so we're working with them to try to test out the multivariable predictor in a prospective setting. And as you say, that's the sort of evidence you'd need probably to take it to the next stage, either get start to think about approvals, or continue to get further industry funding.
Absolutely.